I mean, DNA electrophoresis all semester, so they know pretty much about DNA and Agarose electrophoresis. So I use that as the jumping point for talking about protein electrophoresis. I start with what they know about DNA and DNA electrophoresis and go on from there. Um, that's sort of the way I'm going to start this webinar. And as Julie said, if you have any questions, just type them in. We'll try to get to as many as we can at the end in the text chat. So, all of you know the central dogma of molecular biology: DNA, RNA, protein, and that gets to what you see. And proteins are what determine the structure and function that you see around you. And proteins, I like to say proteins are the point. I mean, DNA gets a lot of attention, but proteins are what really make the difference. And in the picture here on the right, this is a bunch of, these are all flowers in the sunflower family. And as you can see, they're vastly different in colors and shapes and things like that. And the DNA in all of those is almost all the same. It's the proteins that make the difference. And I'm a dog person, so I always like to point out that all dogs are in the same species, yet obviously they're vastly different in appearance, and the DNA is almost all the same, but the proteins are very different, and that's why you see the differences that you see. So I'm sure most of you have done DNA electrophoresis before, so let's talk a little bit about it, or about electrophoresis in general. The word origin is from Greek, and it's electro for electricity, and phoresis means basically carried by. So electrophoresis means, sorry, I'm having trouble getting into advance. Electrophoresis means literally carried by electricity. And it's been around for a long time. It was actually over 200 years ago that a German scientist who was actually working in Russia discovered that clay particles in water would migrate toward an electrode. And so it's been around for quite a long time. And if we were to duplicate that, if we put a bunch of charged molecules into a solution, what you would see, and then we applied a current to that solution, what would happen is that the positively charged molecules would migrate toward the negative electrode, and the negatively charged molecules would migrate toward the positive electrode. But in solution, that's not very helpful, unless, of course, you're electroplating something, in which case it would be useful. But for us as scientists, that's not really very useful. So what we do when we're doing electrophoresis of DNA is we put a matrix in the molecules that we want to separate go into a matrix, and for DNA we use Agarose as the matrix, and it separates the molecules according to size. So if this is the top of our gel and the samples are loaded up here, the smaller the molecules, the more easily they can migrate through that matrix. So you get the smaller molecules at the bottom, and the larger DNA molecules are going to be at the top. So what do we know about DNA in general? Well, we know quite a lot. We know that DNA is a polymer of 4 bases, and that those polymers are hydrogen bonded together to form a double helix. And so the hydrogen bonds here, the bases here, they're going to be base paired. Here's C and G base paired together, and A and T base paired together. So basically, it's 2 long strands that are held together by hydrogen bonds. We also know that they're negatively charged because it is deoxyribonucleic acid. So all along here on the outside of the DNA helix, you've got negatively charged groups. So when you put a DNA molecule in an electric field, it's going to migrate toward the positive electrode. The other thing we know about it is that the average base pair size is about 660 Daltons. Now, when you talk about DNA, usually we just talk in terms of base pairs. In other words, if we have, we just say our standards and everything are just X number of base pairs. But it actually, there's another measurement we use a lot, and that's the Dalton. The Dalton is approximately the mass of a hydrogen atom, or in other words, the mass of a proton. And a DNA base pair is approximately 660 Daltons. And we'll talk a little bit more about that later as we get through the talk. All right, so what do we know about protein molecules? Well, do we know much about their size or their charge, composition? Yeah, we know quite a bit. And protein composition, we know that proteins are comprised of amino acids, and there are 20 standard amino acids and then a few other non-standard amino acids. And the drawing here over on the left is a DNA molecule. Is a protein, is like a schematic of a protein amino acid. And the R group, which is shown down here at the bottom, is what makes each amino acid different. So it's consistent. Each amino acid has an amine group at one end, a hydroxyl group at the other, and then it's got this R group in the center. And that's what's going to determine the characteristics. So if you look over here at the graph, the table on the right, you may have nonpolar, polar, some of them are acetic, some are basic. Some contain aromatic rings, and there are others that contain sulfur molecules, particularly those are methionine and cysteine. And we'll talk a bit more about cysteine. It's very important, as that's the amino acid that's responsible for doing the disulfide bonds between between or within proteins. So here are a few examples of amino acids. Here up in the top, here's our schematic again. And now with the R groups down here, you can see here's cysteine that I just talked about. So its R group is a CH, and then there's the sulfur molecule. Histidine over here we'll talk a bit more about that. That's an important molecule too. Phenylalanine has a big group on there. There's a phenol ring. And then alanine. Some of them are very simple. Here's alanine. And the alanine has just a CH group on it. There is actually one that's a little smaller. Glycine has only a hydrogen ion there, and it's the smallest. It's only 71 Daltons. And the largest amino acid is phenylalanine, tryptophan, which is 204 amino acids. So what happens if we put a bunch of proteins in an electric field? And this is the same image we talked about earlier. We've got positively charged, we've got negatively charged, and depending on the pH, we may have neutrally charged proteins in there as well. So when we apply an electric field to that, you're going to get the same result. Your positives are going to go to the negative electrode, your negatives are going to go to the positive electrode, and those that are uncharged at that pH are going to just hold still. So what do we know about protein structure? All right, so we know with DNA you've got a string of bases and they are in a double helical form. Well, proteins are a bit more complex. We've got primary structure, which is just the strand of amino acids, what order they're aligned in. Secondary structure will be beta pleated sheets and alpha helices, just like the alpha helix is just like in DNA. The tertiary structure is how those are folded together, and then you may even have quaternary structure. If you have two polypeptide strands, they can actually be folded together to give you a quaternary structure. One other thing we have to worry about with proteins is disulfide bonding. I mentioned that the cysteines can form disulfide bonds either within or between polypeptide strands. And in this example, you can see the red bars represent disulfide bonds, covalent bonds connecting two cysteine molecules. And you can see one there and one there. So this strand of the protein is attached covalently to this part of the protein. So these can be within a polypeptide or between two different polypeptides holding them together, or both. So that's something else we have to that makes them different from DNA molecules. So what about size? I'm showing two proteins here. The one on the left is TRPCAGE, which is the smallest known naturally occurring protein. It's actually from the saliva of Gila monsters, which I think is sort of interesting because who would be looking in Gila monster saliva for anything? So it's about 20 amino acids, only 2.1 kilodaltons or 2100 Daltons. Whereas the one on the right is Titin. Titin is a muscle protein, which is actually about 27,000 amino acids. It's very big, so it's about 3,000 kilodaltons. So there's quite a range in protein size. So when you want to talk about protein electrophoresis, you've got a number of considerations that you didn't have in DNA electrophoresis, or that you don't have in DNA electrophoresis. The size, the structure and conformation, and also the charge. So let's talk about these one by one. So first of all, if you look at the size of the protein and want to compare it to the size of DNA molecules, the average base pair, we said, is about 660 Daltons. But the average amino acid is about 110 Daltons. An average protein is about 300 amino acids, which would make it 33 kilodaltons. So that's the DNA that you're going to be talking about. If you want to know how much DNA it takes to code for a protein of average size, we can actually make a rough calculation of that. If you look down at this, this is just a DNA to mRNA, the central dogma. Down at the bottom we've got the amino acids, and you know that it takes a codon, 3 base pairs, to code for that amino acid. The other thing you know is that, so if you just work with that number, you've got 3 base pairs, 300 amino acids. That would mean it would take 900 base pairs just to code for the primary structure of this protein. But of course there's a lot more DNA involved in that. You've got to think about promoters. Some proteins have propeptides that are coded on them. There may be post-translational modifications. So it's going to be a much bigger piece of DNA needed to code for that protein. But the minimum would be, if you go with that 900 base pairs, 660 Daltons, you're talking 600 kilodaltons of DNA to code for a 30 to 35 kilodalton protein. So the way we deal with this, working with the size, is that we use a different matrix. You all know that we use Agarose usually for separating DNA molecules by size, and I like to think of Agarose sort of like Jello. It's, you heat it up to dissolve it, you pour it into a mold, and it hardens in the mold, and then you can run your gel. And if you go back and you now heat that again, it'll dissolve again. You go back to a liquid state. Polyacrylamide is the matrix that we use for proteins in most cases, and it's not like Jello. It's when you form a protein gel, an acrylamide gel, it's more like a plastic. It undergoes a chemical reaction that's not reversible. You can't go back and melt a DNA gel, an acrylamide gel, and get back to what you started with. So I said, oops, sorry. Although in most cases we use polyacrylamide to separate protein molecules, you can use Agarose. This is an example of running fish proteins on an acrylamide gel and an Agarose gel, and you can see there's a big difference. You get much sharper bands and much more resolution with the polyacrylamide, but you do get separation with Agarose, and you can, if you're looking for changes between different samples, as in this case, you can see, for example, there are differences in the protein profiles between, say, shark and salmon if you look down in this area. So it can be used. You get a much better sample, much better resolution if you use polyacrylamide. All right, so let's talk about polyacrylamide for a moment. Polyacrylamide is prepared by mixing acrylamide monomers with a cross-linker called bisacrylamide, and then you add a catalyst and the gel, pour the gel in a mold. Just in that respect, it is like DNA electrophoresis, and the gel forms. So the molecule that you're looking at here is acrylamide. It's a very small little molecule, and when you add the polymerizing agent, it forms a long polymer. So here are the acrylamides down here forming a long polymer. The other molecule I talked about, bisacrylamide, is the cross-linker. So this is bisacrylamide here, and what it's doing is it's forming a cross-link between this long strands of acrylamide. So this is forming a matrix. Just like with DNA electrophoresis, you choose your percent acrylamide that you need for the molecules that you're interested in separating. For example, if you're doing DNA electrophoresis on plasma DNA, you might be using 0.9 or 1% acrylamide gel. And when you're doing something like looking at a PCR product, you might want a 3% agarose gel. So you do the same thing with acrylamide. You base the percent acrylamide that you used on the molecules of interest that the ones you want to look at. And this is a graph I like to use that came out of a book. The y-axis is size in kilodaltons of the proteins, and the x-axis is the migration in the gel. So for instance, if you wanted to look at a protein that's say 10 to 20 kilodaltons, you'd want a gel that's about 15% acrylamide. If you wanted to look at something larger, say a 100 kilodalton protein, you might want a gel that's 5% acrylamide. The higher concentrations, just like with Agarose, are used to separate smaller molecules. And there's a good way to look at this. This is actually a graphic looking at the same molecular weight standards in gels of different concentration. So if you're talking about looking at something, the numbers I know are a little hard to read here, so I've got red arrows in marking a 45 kilodalton standard, which is here. All right, in an 18% gel, it's going to be way up in the gel. But then as you move over, say at 10% or 7.5%, it's moving further down. At a 5%, that protein is going to run off the bottom of the gel. So if you're interested in looking at a protein that is, say, in the 30 to 45 kilodalton range, you would want to use a gel that is, say, 12 to 18%. You certainly wouldn't want to use a gel that's down at 5 or 7.5%. Okay, what's the next thing we need to talk about? How about conformation of proteins? As we said with DNA, to denature DNA, any of you that have done PCR know, all you have to do is to heat it in the first step of PCR and the molecules denature. The hydrogen bonds break. Proteins are a little more complicated, which is why I love them so much. They have secondary, tertiary, and quaternary structure that if you don't deal with those, they're going to confound the separation by size in the gel. So the way we deal with this is to place the samples in a sample buffer that contains reducing agent and detergent, heat the samples, and that reduces and denatures them. The disulfide bonds are reduced by adding a reducing agent, and the two I'm showing here are beta-mercaptoethanol, or known as BME, and dithiathreatol, which is another reducing agent. Both of these, down here I've got a little graphic of the two cysteines held together by a covalent bond between the two sulfurs. And these reducing agents reduce that bond, so you're left with two sulfurs with hydrogens on it. So this is going to disconnect the strands that were held together. One little interesting note on this with beta-mercaptoethanol, beta-mercaptoethanol smells like rotten eggs. It's pretty nasty. Dithiathreatol smells a little better. If you've ever had a permanent on your hair and that nasty smell, what you're actually doing is using a reducing agent to break all the disulfide bonds in your hair, and then when they put it in curls and wash it out, they're allowing those disulfide bonds to reform in the shape that you want. So just a little side note, if anybody ever says practical uses of science, there it is. The next thing we'll talk about is that there is a detergent in the sample buffer, and the one that is normally used is SDS. It's sodium dodecyl sulfate, and this is a really commonly used detergent class. If you look at anything in your household that suds, it'll have either sodium dodecyl sulfate or sodium lauryl sulfate, something like that in it. So it's an ionic detergent. There's this nice big charged O at the end. And what this does is it disrupts the non-covalent bonds, for example, the hydrogen bonds within the molecule. So in the presence of heat, this is going to disrupt and basically denature the proteins and get rid of that secondary and tertiary structure. The other really cool thing that this does is it also deals with the issue of charge. Remember we said that proteins have either a positive charge, a negative charge, or depending on the pH, no charge at all. So you have to do something so that they will migrate to the positive electrode and separate by size. And SDS plays a role in that. If you add SDS in your sample buffer, it's going to coat the denatured proteins, and you can see it over here in this graphic at the right. You've got your coily proteins, and once they're denatured, they're coated with these negatively charged SDS molecules. And what makes this work so well is that SDS binds to proteins at a fixed ratio. For every 1 gram of polypeptide will have 1.4 grams of SDS bound to it. And these are strongly negative molecules. That O- group on it makes them very strongly negative, so that that negative charge will actually outweigh any intrinsic charge or lack thereof on the protein, making them all negative. And since the SDS binds at a fixed rate, they're all the proteins, all the polypeptides are now going to have the same charge to mass ratio. So what this means is when you do electrophoresis, they're going to migrate according to their size. They'll all be negatively charged. One interesting thing about this is something that came out from a scientist named Lamley. This paper on this came out in 1970, and that's where we get the name Lamley sample buffer, is because he developed this buffer. And the paper he wrote was published in Nature, and it had something like 130,000 citations as of last time I looked. So it's definitely a commonly used technique. All right, so what's in our sample buffer? Well, we've talked about most of these. Well, there's a general buffer. There's tris. We've talked about the detergent, a reducing agent, and then just like with DNA electrophoresis, there's going to be glycerol to keep the sample to make the sample denser than the buffer, so that the sample won't just float away into the buffer. It's going to stay in the well, and also a tracking dye. And usually the one we use for this is bromophenol blue. So the tracking dye, just like for DNA electrophoresis, serves two purposes. One is it helps us load. It's always easier to load something if you can actually see it. And the other is it gives us an idea of where the samples have run in the gel. This is just a little cutout of a gel showing a dye front here of the bromophenol blue, and it runs at about 670 Daltons. So generally your proteins are all going to be lined up behind it. So you know if you've run that dye front down to the bottom of the gel, your proteins will be stretched out behind that. So now let's talk a little bit about we're going to be running vertical gels, and this is very different than agarose electrophoresis. When you run a DNA gel, you dissolve your agarose, pour it in a mold, let it set, remove the comb and pour buffer all over it and load your samples. Well, the gel is actually flooded in buffer. There's buffer on the surface too that will carry some of the current. The gel will not carry all of the current. With protein electrophoresis, it's different. The gel itself is going to carry all of the current. It is the only connection between the two electrodes. And this is just a gel box. This picture here is a gel box with the lid off. You can see right where I've got the star now that that's where the wells are. And behind that, in this space, is the upper buffer chamber, which contains the negative electrode. The gel is running down between two plates to here, and down at the bottom here is the other electrode, the positive electrode. So the only connection between this chamber and this chamber, which contain the two electrodes, is the gel itself. So how are we going to get the samples to all enter that gel at the same time? This is my little graphic. This is a cute little well at the top with two identical proteins in it. As you apply a current to that, you will see that they will each of these little proteins are going to migrate a certain distance into the gel. Well, if you just turn on the current and one is at the top of the well and one is at the bottom of the well, they're not going to be at the same place in the gel at X amount of time. And so there's a really neat system that has been developed to deal with this. And actually this was developed 50 years ago, which I think is pretty cool. Ornstein and Davis published this in 1959. I'm not going to get too much into the science, although there's some references at the end, and I'll be happy to answer questions because this gets a little too complicated for the scope of this talk. But the nutshell version is there are two gels, one on top of the other. The top is the stacking gel, the bottom is the separating or resolving gel. They have different pHs. The stacking gel has a lower pH, it has a lower molarity of buffer, and it has a lower percent acrylamide. The resolving or separating gel that's the one that will behave the 12 or 15 or 18% acrylamide. It'll have a higher molarity and a higher pH. And what this does, and the long and the short of it is, if you load proteins up here in the well, by the time they reach this interface between the two gels, they're all going to be lined up in a straight line, and we call that stacking. So here's an example in a photograph taken of a gel. You can see the wells here at the top, and here are the proteins. You can see they're sort of spread out with the dye as the proteins migrate through the gel. And then in a short amount of time, you can see that they've stacked. You can tell that it's visualized by the stacking, by the loading die, this nice blue line. The proteins are stacked up like that. So by the time they hit that interface of the two, they're all going to be entering the separating gel at the same time. Okay, so we're ready to go. So we've got our gel box assembled, we've got our samples ready, and we're ready to load our samples. And so what we do here is, just like with DNA, we're going to run a size standard alongside our samples, our experimental samples. And I've got a couple of examples here on this one but actually showing both new technology and old technology. Those of you that have used size standards, which I assume is most of you, for DNA electrophoresis, you know that a lot of you, if you've been around a while like I have with this stuff, know that you might use a Hindi 3 digestive lambda DNA as a standard originally, and now you may use a DNA ladder that has things that are very, at stepwise intervals, every 100 bases, etc. Well, protein electrophoresis is the same way. The original standards were using molecules of known molecular weight, BSA, ovalbumin, etc. More modern standards are like ladders. They are recombinant proteins at very particular set molecular weights, so you can have more of a ladder effect. And these are sort of cool. The ones on the left, you have to stain like you do your gel, and I'll talk about that in a minute. The ones on the right are pre-stained, so you can actually see them running in the gel, so it's pretty cool. So running the gel, most gel boxes will run from 1 to 4 gels concurrently. This is the Tetracell shown over here on the right, which is a Bio-Rad gel box, and it'll run from 1 to 4 gels. You can either prepare or purchase your gels and buffers. For the gels, I really recommend buying them, because acrylamide, you can pour your own gels. Acrylamide is a neurotoxin. Neurotoxins are generally no-nos in a high school classroom and maybe even in a college classroom. I don't pour my own gels now for my class. I generally buy them. Buffers the same way. You can make them or you can buy them. The average running time for a gel like this is less than an hour, usually 30 to 45 minutes for a gel, one of the gels of the current sizes now. So we're running our gel. Our next thing is, how are we going to see these proteins? Except for a few proteins like cytochrome C, we don't have any way of seeing them in the gel. So we have to use some kind of stain. And generally we use a stain for total protein. You soak your gel in stain, and then you destain it to remove any non-specifically bound stain. And what you get with are blue, well, in the case of this example, are blue bands on a relatively clear background. And historically, most of the stains that have been used have been Kumasi based, in which case you do have blue on clear. And again, historically, most of the stains that have been used have used a form of Kumasi called Kumasi R, which is, that's been the historic stain of choice. The downside on it is that it has to be dissolved in organic solvents. It has to be dissolved in like methanol and acetic acid, so again, can be problematic in the classroom. More recently, more stains are being developed that use Kumasi G, which does not require organic solvents. It can be in an aqueous solution, so they're a bit safer for classroom use. The second function of this stain is actually it fixes the proteins in the gel. So whichever you're using, whether it's a Kumasi blue, historically, Kumasi blue stain, or a modern stain like BioSafe, which is a Kumasi G stain, both will serve to fix the proteins in the gel so they won't just like wash out. So what can we tell from an SDS-PAGE gel? Here's a nice gel on the right that actually shows a protein purification in different steps. So I'll just mention what the, excuse me, I seem to have lost my mouse. Well, I'll keep talking even if I have lost my mouse. I don't know what's going on here. Julie, you can hear me all right, right? Okay, I'm just going to keep going, and you may have to advance the slides if I ask you to, because it appears everything is frozen up on this end. Okay, so here's the gel. Lane 1 is the standards. Lane 2 shows the gamish of proteins, and the arrow marks the protein of interest, which is about 50 kilodaltons. And am I back? Oh, thank you, Julie. Never mind. Okay, third lane is, so what this is a purification using chromatography, and the protein's been loaded on a gel, and then lane 3 shows the sample that what runs through the gel. Lanes 4 and 5 are washes, and then lane 6 is the protein that's eluted. So what you can see from this, there are a couple of things you can figure out. One, the size of the protein. You can see that the protein of interest is running right about with the 50,000 marker, and you can also see the number of proteins. In lane 2, you can tell there are a lot more proteins present than in, say, lane 5 or lane 6. So you can talk about the number of proteins. You can talk about the quantity of proteins. Sorry, I think I'm back. Maybe I'm not. The quantity of proteins. You can tell if you compare two side-by-side bands, you can tell that there is more in a darker band. But that's also assuming that the dark band contains only one protein at that molecular weight. There could be 5 that happen to be at exactly the same size. You can talk about purity of proteins. You can tell that that lane 6 is a lot purer than the sample was in lane 2. What you can't tell from this is protein identity. You can't. There's no way to definitively say that any one protein in there has that you know the identity. You can make pretty strong guesses, but you can't say it definitively. Here's another gel, and this one is a fish protein gel. I like this one, and you can get suggestions from. You can have very strong suspicions of what your proteins are. For example, if you look over on the right side of this gel, you'll see the actin standard that's run, and it's about 42,000. And you can look in the other lanes and see that there is another protein that is about 42,000. So you've got a pretty good idea that that is actin at that position. You just can't say it definitively from this. The other thing that this one picture demonstrates is that you can look at changes in protein profile. These are different fish. You may compare the salmon lane, which is lane 3, with the trout lane, and you can see they're very similar. Well, salmon and trout are closely related. That might explain it. But then compare that to, say, catfish, and you'll see an awful lot of differences in the protein profile. So that's something else you can tell from looking at these gels. So what do we do if we want to confirm an identity? Well, sorry. Oh, my mouse is back. Yay. All right. I'm back, Julie, I'm back. All right, so what we can do is a Western blot. And what you do with a Western blot is you transfer your proteins from the gel to a membrane using electricity. And usually you can use the same gel box that you used for running the gel. You apply a current, and all of your proteins that are on that gel move to the membrane. And down and below, the second picture is a picture of the gel, a graphic of the gel, and those little lines are supposed to be our proteins that have transferred. And here we've used a nice colored standard, a pre-stained standard, which is good because you can confirm that your proteins did transfer. All of the spots in between these proteins, though, need to be blocked so that you don't get nonspecific binding when you probe this membrane. Generally, what I use for blocking is something I make with carnation non-fat dried milk. Just a solution of that, and all of these spots get blocked with milk proteins. So once you have your proteins blotted onto the membrane, you're now going to probe for your protein of interest. And here's a great little graphic. I don't know how many of you have done, I assume some of you have done immunoassays like the ELISA assay. Well, the principles are exactly the same in Western blotting. Say our little purple triangle is our protein of interest. Say it's myosin, a muscle protein. We probe the membrane. All right, let's see, the little red guy is something we're not interested in, and these little circles are actually blocking agents. So they're they're the proteins that are blocking the unused sites on the membrane. So we probe with an antibody that recognizes, for example, myosin. And these antibodies can be either polyclonal or monoclonal. And again, I'm not going to go into too much detail here because they are, that's a talk for another whole webinar someday to get into the immunology of this. But you can prepare or buy antibodies that recognize the proteins you're interested in. So after that binds and you wash off anything extra, you then probe with a secondary antibody. And that secondary antibody will recognize the primary antibody. So for instance, if this is a mouse anti-myosin primary, this might be a goat anti-mouse IgG that would recognize this. The other cool thing is that you can get an amplification here. You can have multiple secondary antibodies binding to one primary, even though it shows only one here. The secondary antibody is also tagged with an enzyme tag. These can be, they're two different, the one I'm used to using, one of the common ones is horseradish peroxidase. Another one that's commonly used is alkaline phosphatase. And that tag is then, you use a substrate that will either develop a color, say for instance a color on the membrane, or there are also some chemiluminescent ones that you can detect with a phosphoimagers, things like that. But generally it's going to be like a color development, and you'll see, say, a colored band on a white membrane. And I'll show you an example of that in just a minute. Well, here's the example. The Comparative Proteomics kit, which have a protein profiler and Western blot modules, use our favorite fish proteins and solubilize them in sample buffer, analyzed by SDS-PAGE, and then in the protein profiler module you look at total protein staining, and in the Western blot module you electroblot and probe for myosin light chain. So here's an example, another fish gel. I don't know, you may be getting tired of fish gels, but they're pretty cool, showing, again, the standards and the different fish samples, an actin-myosin standard, and in this case we've marked actin and we've marked the myosin light chains. If you then transfer this to to membrane, and then probe for myosin light chain, over on the right you've got our blot, and the brown bands here, they should be a little purplish, I mean the color's not quite right, are the myosin light chains. So now you've definitively identified that as the protein that you're looking for. The other thing sort of interesting on here is here are the colored standards that both give you your sizes, and they also confirm that your proteins transfer to the gel, because if you can see the markers, you know that the proteins are there too. There's one other really neat example. There are some proteins, particularly GFP, which is green fluorescent protein, which some of you may have used, that will show up on a gel without any staining. This is on the right side, we've got a gel stain for total protein, and the green circle here actually marks the protein band, stained protein band of GFP. But if you take that same gel and put it under UV light, you can see the GFP in the gel. And the standard in this case also has two bands that'll show up under UV light. So I just think that's pretty neat. In this case, the GFP has been denatured but not heated. If you heat it, it denatures, it will completely come unfolded, and then you can't see it on the gel. But if you just do processing with reducing agent, then it still shows up. So I think that's pretty cool anyhow. All right, let's talk about doing this in the classroom. What do you need? Well, you need a gel box, at least one. You need a power supply, again, at least one. And the same boxes that you use for DNA electrophoresis, the same power supplies, will work for protein electrophoresis in most cases. You need pipetters for preparing your sample and loading your sample, and you need gels and buffers. When you're doing this in the classroom, I know everybody's got different time periods, different class periods, so it's going to be really dependent on that, and whether or not you've got kids that are very cooperative or not, because some, as you know, some are and some aren't. If you've got a shorter time period, the good thing about this experiment doing protein electrophoresis is there are a lot of breaking points. You've got after you can prepare the sample samples one day, load and run the gel the next, and discuss the results another day. Or if you have a block scheduling, say with 2 hours, you could actually do all this in one lab period. One of my favorite things about this experiment is that you can let the students plan their own experiments. Sample choice can be a lot of fun. You can tell them what to do, or what I like to do when I'm doing this in a workshop setting particularly is have a selection of samples available, give them a list and say, okay, what questions can you ask and answer using this technology? What will the proteins tell you? So I might have a list of 10 different samples, so it might be I might look at have an example on there of processed versus unprocessed food. So I might have smoked tuna, fresh tuna, and canned tuna. They might choose that. I might have fake crab in there, and they might be trying to figure out what's actually in the crab delight. You can ask evolutionary questions. Are things I mentioned briefly that salmon and trout are very closely related. Well, you might want to try to see what other fish are either closely or distantly related from this. One I don't think I got this to work, but sedentary versus always moving. What if you compare a fish like a flounder to something like a shark that's always moving around? Would there be any differences? The cool thing is you can let them figure out what they want to ask, and they can come up with some rather original ideas on things to ask. Obviously, in a classroom, you want to try to eliminate as many safety hazards as possible. One thing is, of course, use precast gels. If you can avoid using neurotoxins in the classroom, that's always a good thing. Use a stain that doesn't require organics. Use something that's like one of the BioSafe, that you don't have to have the methanol and acetic acid. Current research equipment, most of what's out there now, is designed to prevent shock hazard. So if the lid comes off, you no longer have a current and there's no danger of a shock. One other possible way is you can eliminate the reducing agent. These are a couple of gels I ran, again looking at seafood samples. And the top gel is with beta-mercaptoethanol, the reducing agent, and the bottom was without. And you can see there are differences if you compare, say, the proteins here without beta-mercaptoethanol and here with. You can see clear differences, but it still would work if you want to compare. You can still see differences in the protein profiles. It wouldn't change a result in an experiment like this. There are other experiments, though, where it would make a huge difference. This is an experiment showing the purification of antibodies of immunoglobulin molecules, and on the left you've got the samples reduced, and on the right the same samples non-reduced. You can see there's a huge difference in the way they appear here. And one other thing to remember, if you're doing Western blotting, in a lot of cases the antibodies won't bind to proteins unless they're reduced. So you definitely want to think about using a reducing agent if you're doing a Western. So problem areas I've come up with, pouring gels. Besides the fact that acrylamide's a neurotoxin, it's also pouring gel. It's not as simple as pouring the jello of an Agarose gel. There's a technique involved, and it requires some practice. It's just not quite as straightforward. So that can be an issue. Loading the gels. It's sometimes very hard for the students to see where the samples go. Here's a picture of a gel over here, and you can see where I've got that sample loaded, where the wells are. It's sometimes hard for the students to figure out where to put the pipette tip to get the sample in there, and it helps if you have a gel box that has a loading guide like this one does, that sort of directs their tip down into that well. Let's see, reducing agents, as we talked about, sorry about that. Leaking gel boxes can be another issue. Occasionally you have a time when the upper chamber leaks. A slow leak is not really an issue, and as you get better equipment out there, which is like real research grade equipment, that's really not as much of a problem anymore. Another problem I've run into, and this when teachers call me asking about this, one of the biggest things that that people don't understand is that center buffer chamber I talked about. You have to fill that chamber so it's above the level of the wells, or you won't get electrophoresis. You also want to make sure that the samples run far enough into the gel. Students have a habit of seeing that nice blue line about halfway down and think, oh, that's good enough. Well, you need to run the gels as far down as possible so that you get a really good separation. If you stop the gel after, an inch or so, all your protein's going to be spread out in a very small area. Whereas if you run the gel all the way to the bottom down here, you'll get much better separation. And then my favorite, I always have like blue stained fingers because I don't follow my own advice. You look like a Smurf if you use any protein stain and don't wear gloves because we have a lot of proteins in us. And yeah, I have blue fingers a lot of times. So I think that's the end here. There's my contact information. And the Explorer and Bio-Rad, if you have questions specifically for Bio-Rad, the Explorer website, and the reference I talked about, if you're interested in the stacking gels. Okay, this bulletin, which you can download from the Bio-Rad website, the bulletin 2144, has a really good explanation on stacking gels and how they work. Like I say, I used to say that was a prelim question. It's not something to really go into in a talk like this. And finally, the references at the end, the ones I used for preparing the talk, and I'll be happy to help with any questions. Thank you.
Thank you, Kristi. So if you would take a look at the chat window and read aloud some of the questions that are there for the audience and give us your answers, that would be great.
Okay, let's see, where's the first one? All right. Reference for preparing Ken, do you mean preparing the fish samples? Yeah, it's very easy to do. Basically, you cut up a little piece of fish and put it in the sample buffer. I'd be happy to send you the protocol if you want to email me, or you can actually, I use basically the same one that Bio-Rad does, and so if you download, you can download their manuals or their PowerPoint, and it gives you the directions. But basically, you cut off a small piece of fish and let it sit for 5 minutes in the buffer, and that solubilizes enough of the proteins in the fish. You don't want to chop the fish up, I'll tell you that. Non-denaturing conditions. Eduardo, do you mean non-denaturing? You can prepare your sample buffer, if this is what you mean, without reducing agents. But a non-denaturing gel, all in all, is a whole different ball game. Could you elaborate on your question? I'll try to get to it. Yes to the PowerPoint in your classroom. I believe you can check with Julie. Rob, you had the same question. Let's see, Edward. Please repeat the full question. Thank you, Julie. I didn't get down to that part. All right, let's see. Ken had requested how to prepare fish sample standards, and I said basically solubilize them in the buffer and either get the procedure from me or from the Bio-Rad website. The is easy. Non-denaturing, how to do the procedure for non-denaturing conditions. If you want to just not reduce your sample, you just don't put the reducing agents in there. And you can also, play around with heating or not heating depending on what your samples are. Accessing the PowerPoint, I believe Julie's already answered that. Eduardo, yes, Eduardo asks, when you want to run a gel without denaturing, that's a little more complex. You can run not denature your samples and run them in a standard gel without problems, but running it as a totally non-denatured, as a native gel, is a whole other topic, and I don't think we can get into it here. I'd be happy to answer questions outside of here. Plant samples. Barbara is asking about running plant samples on this. I believe my colleague has that on her website. If you email me, Barbara, I will try to find the link for running that and send it to you, because I'm not a plant person. if Karen asks if base pairs are 660 Daltons, why are amino acids fewer Daltons? If you look at a base pair, you're talking, look at the number of atoms that are actually there. You've got the sugar groups and look at the structures. Relatively speaking, the amino acids that I showed you, you saw the simple carbon with the amine group on one end and the carboxyl group on the other, and then the R group that could be as little as a single hydrogen. Compare that to what's in DNA. It's deoxyribonucleic acid. There are a lot more atoms there. If you just compare the structures, you'll see what I mean. I hope that answered your question. Kathy's asking how do we access the PowerPoint. Julie's going to send out a link for that. Patricia asks, in the slide for of the green fluorescent protein, what were the two fluorescent standards? No, that was not a native gel. It was a standard gel purchased from Bio-Rad. That is actually part of the After P-Glow experiment that you can also download from the Bio-Rad website. But the kaleidoscopes, I think it's Julie can correct me on this. It's the hang on, I'm looking. Kaleidoscope standards have a Precision Plus Kaleidoscope standards have the two bands that also fluoresce under UV light at the 25 and the 50. So they're in there. And no, it was not a native gel. Eduardo asked about sensitivity related to stains. Well, you can look up, I don't have the numbers off the top of my head for Kumasi stains. There are other stains that are more sensitive, like silver stain. There are also stains that are fluorescent. I'd have to look up the exact numbers. With Kumasi, I don't remember off the top of my head, Eduardo. I'm sorry, I'd have to look that up. Cheryl wants plant protein information. I'll be happy to send it. Just email me. I'll send you the link for that. Will the PowerPoint have the voice? I don't know, Julie. I assume there will be a voiceover somewhere if anybody wants to listen to my voice today. Oh, thank you. Armand says that 50 nanograms is the for Kumasi is the limits of detection. I was thinking it was down in that range. Hey, Kevin. All right, I'll send a link for the protein protocol to Julie, who can put it with this. My colleague Erin Dolan, I think, has one on her website, so I will look for it. Oh, okay. All right. So are there any other questions?
Thank you everybody for listening today, and if you have suggestions for future talks, we welcome your ideas. If you could complete the survey that is offered when you exit, there's a place for you to provide us feedback on what you liked or didn't like today, and also future topics you might be interested in. So thank you very much for participating.